In this experiment, you will be digesting 2 clones of pyruvate dehydrogenase. You'll be using restriction enzyme, which are enzymes that specifically cut DNA. Restriction enzymes recognize a specific sequence on the DNA and then cut it. Restriction enzymes are naturally found in bacteria, and they're used to digest foreign DNA that is injected into them from phages or bacteria viruses. Restrictions enzyme recognize specific sequences such as the one shown here. They can be from 4 or more bases. The majority of the sequences recognized by restriction enzyme are palindromic, which means they can be read from either directions. Many enzymes cut the DNA in a staggered manner, leaving either a 5' overhang, such as ECAR1, or a 3' overhang. However, some other enzymes cut the DNA, leaving just a blunt end. You will be provided with plasmids. Plasmids are circular DNA sequences naturally found in bacteria. In nature, they divide independently from chromosomal DNA and often carry genes that protect the bacteria from antibiotics. In genetic engineering, it has been possible to use restriction enzyme to cut pieces of DNA of interest and inserting them into plasmids, and then make many copies, clones, in bacteria. You will be looking at 2 cDNA clones of PDH beta subunit. One of it contains 11 exons, and the other one is a splice variant in which exon 6 is not in the sequence. You will have 2 plasmids containing the 2 clones, PDH-B1 and PDH-B2. They're both being cloned into the BAMH1 site of the PCX plasmid, and note the 2 different sizes of the inserts. In your protocol, you've been provided with the restriction maps for the 2 clones, PDH-B1 and B2. From these maps, you'll be able to design your restriction digestions. You'll be able to analyze the product of your digestion using gel electrophoresis. Gel electrophoresis is a system whereby fragments of DNA travel in an electric field towards the positive pole because DNA is negatively charged. In the example shown there, the plasmid cut with BAMH1 will produce 2 bands, while the plasmid cut with BAMH1 and ECAR1 will produce 3 bands. From the standards, you'll be able to recognize the size of those bands. To set up your digestion, you have to consider the amount of DNA you need, which is about 5 micrograms, the specific enzyme buffer. Each enzyme uses a different buffer. Water to add to make up a specific volume of 30 microliters. And then the enzyme, 1 or more, that you want to use in that particular reaction. Be aware that enzyme is stored in 50% glycerol, and you must bring that concentration of glycerol to less than 5% in the reaction. So your task is to design 2 restriction digests that will clearly identify the 2 plasmids that you're using. So to set up your digestion, you need to first of all study the restriction map and select the enzymes that you think are going to be most useful. There is more than one possibility, so it's really your choice. You will then set up your digestion, incubate it for 20 minutes at 37 degrees C, and then add the loading buffer. You can then load that onto the agarose gels. Because of time restriction, we will run the gels for you. This practical does not involve compounds which are particularly dangerous, but do follow standard procedure for safe work in the laboratory. Remember that you have to complete your quiz before you can come to the lab.